So in the last lecture we looked at some of the methods that neuroscientists have been using to manipulate genes, and we saw some of the early experiments that used this technology to try and test the synaptic plasticity learning hypothesis. And in the next couple of lectures I'd like to introduce you to some very recent techniques that are really transforming how we approach this problem. And the two techniques are called optogenetics and chemogenetics. So this idea has been around for a while, and this is an extract from a quotation from Francis Crick, who was one of the co-discoverers of the structure of DNA, a very insightful thinker, and he wrote in 1999, a great need in neuroscience is to be able to turn the firing of one or more types of neuron on or off in the alert animal in a rapid manner, so kind of like natural control of an animal in a way. The ideal signal would be light. This seems rather far-fetched, but it is conceivable that molecular biologists could engineer a particular cell type to be sensitive to light in this way. So that's exactly what happened. So recall that neurons are controlled in their action via their receptors. That's kind of the gateway to their internal state, if you like. And so if we want to control neurons as experimenters, using receptors is a natural way of doing that. We also have been using electrical stimulation directly, but controlling neurons via their receptors is a very powerful means of regulating what happens to them. So gene technology has let us make receptors that are not natural for neurons. And this has been a really amazing development. And there are two broad ways that we've been using this technology. One is optogenetics, and one of them is chemogenetics. So optogenetics is based on ion channels in blue-green algae called opsins. So these are chemicals that sit in the membrane, and they're sensitive to light. And if light shines on them, then they open ion channels, and something happens to the cell. You can see the chemical structure of one of the most important opsins, channel rhodopsin, over there on the right. And obviously blue-green algae are single-celled organisms, and they float around in the ocean or in ponds and so on, and so they're exposed directly to light. Mammalian neurons are deep in the brain. They have no need for opsins. The only opsins that we have are in our retina. But using transgenic technology, molecular biologists have taken opsins from algae and put them into mouse neurons. So this is just to illustrate what happens to an opsin protein if you shine light on it. So they sit in the membrane like any other receptor, and they have an ion channel, and when light shines on them, that causes the protein to change shape, and the ion channel opens and ions flow in and depolarize the cell. Sometimes the ions are positively charged and sometimes they're negatively charged, so they will either positively increase the positive or negative charge inside the cell and depolarize or hyperpolarize it respectively. So this means that if we put opsins into mouse neurons, we make the neurons light sensitive. Now obviously, as I mentioned, the neurons are inside the skull and they never see light, but we can get light into the brain via optic fibers, which are very, very fine hair-like glass fibers which will transmit light. And can transmit light at quite a high intensity, so laser light right down into neurons. So what you can see here is a picture of a mouse that has optic fibers going into the brain, and blue laser light is being shone down that optic fiber. You can see the blue glow. And so it's possible to get light to the neurons of mice that are carrying genes for opsins. So in the experiment shown here, these neurons had the opsin channel rhodopsin, and when the blue light was shone on them, that activated the neurons, and in this particular experiment that controlled neurons in the lateral septum, which mediated how aggressive the how aggressively the animal responded in a threat situation. So as I mentioned, some opsins excite neurons and some inhibit them. The ones that let in positive ions excite them, and the ones that let in negative ions inhibit them. The common opsins that excite neurons are channel rhodopsin, and the common opsins that inhibit neurons are called halorhodopsin. So that was optogenetics. Chemogenetics is a similar principle, but instead of using light, we use chemicals. But again, the idea is that you take a receptor that's not natural for that animal and you insert it using genetic transgenic technology, and then you make that neuron sensitive to stimuli that it wouldn't normally encounter. So with this chemogenetics, what molecular biologists do is to make a receptor whose ligand, so the transmitter molecule that would normally bind to it, the ligand is an artificial one. It's not a naturally occurring one. So this receptor sits in the membrane of the cell and it's not normally activated because the animal doesn't naturally make the ligand for that receptor. But if you then inject the ligand, now you can activate those neurons. So this is a technology called DREADS, designer receptors exclusively activated by designer drugs. So the designer receptor was made by a molecular biologist and the designer drug, the ligand, was made by a molecular biologist. And when you put those two things together, then you can control neurons. So this is an example of how you would do this. So you make the gene for the receptor and then you package it up into a virus, as in the way that we saw in the last lecture, and then you inject that virus into the target region of brain. And also you would typically add into that viral construct a reporter gene of some sort, like green fluorescent protein, so you can see which are the cells that took up that stretch of DNA and now have those receptors. And then at some time of your choosing, so it could be weeks later, it certainly needs to be at least a couple of weeks later, so there needs to be time for the virus to replicate and to transfect the cells. Then if you inject the ligand for that receptor, and a typical ligand is CNO, this one that's shown here, then it will bind to the receptor and activate the ion channels and activate those neurons, or silence the neurons. So you can kind of control the neurons. And the nice thing about this is that by using the right promoters, you can attach those receptors only to cells of interest. So maybe only cells that make acetylcholine or something like that. So in the next lecture, we're going to look at a real-world example of optogenetics with a complex but very beautiful experiment that seemed to suggest where the engram might be stored.